All right, let's start. Hopefully this will work. So right here, what we are going to talk about, that right here we are looking at the overview of the cell cycle first. So good, everyone?
Yeah.
Yeah.
Okay,
Yeah.
perfect. All right. So the first thing that what you all know from your Bio 101 is the M phase, which is the mitotic phase. So right here in the mitotic phase, you have these super fast events that's going on. Just takes a few minutes to complete that. And after that, there is a big gap that you are seeing that we call as an interphase. And it looks like that there is nothing going on in a cell cycle in this interphase, but there are lots of things going on during this phase. So we're going to talk about these two phases in a separate way in detail, but let's have a first overview. So we are starting right here that a cell which is coming out of a mitosis. So when a cell is coming out of a mitosis, it do have the single pair of the chromosomes, and that means it's a diploid chromosome. Say, for example, for us human being, we have 23 pairs of chromosome, 46 chromosomes. All the cells after this division will have these 46 chromosomes or 23 pairs of chromosome, one set of numbers. Now, we are not going to talk about the meiosis, which is a special type of cell division here. We are only going to talk about the mitosis, where is one cell is duplicated into another, and it's an equal division of all the chromosome numbers. So once it comes out of the mitosis, what happened? The cell can go into a stage which is the G1. So this G1, this phase is the gap phase 1. And here the cell is growing and also checking a different types of environmental conditions. And we're going to talk about that. Now here what you are seeing that there is an offshoot which is called the G0. And what can happen in this offshoot? In the G0, some of the cell may decide that after dividing, it will not immediately divide. It will just remain there. And at certain point of time, upon some kind of stimulation or cell signaling, it can come back and start dividing again. And here you can easily correlate that information. So say, for example, a fly embryo, like a Drosophila embryo, each cell will probably divide every 8 minutes. So it comes out of the mitosis, it will quickly go through the cycle and divide extremely fast. On the other hand, you can see that the mammalian liver cell may not divide in a year. So what will happen, that liver cell will come out, go into the G0 phase and do its function. It's a long-living cell and it will just come out from that G0 upon someone's stimulation. Now, on the other hand, say, for example, some great examples for these are your T cells and the B cells, which are your immune cells, have a very, very long life. They live, some of these memory cells, for your lifelong. And if not needed, they will just not come out. So that's about the G0. So the cells can go into a sleeping mode, means that they will not divide. That does not mean that they are not working. These cells are working and they will only divide when it is needed for that particular tissue or the cell to divide. So what is going on in the G1 phase? So in the G1 phase, the cell is growing in size, and also along with that, it is sensing its environment. What we mean by the sensing the environment, that it is checking that whether there is a good amount of nutrients available, if the environmental condition is okay for the growth. And why it is doing that? Because those conditions are necessary for the cell to grow into the next phase. So what is the next phase? The next phase, what will happen? The DNA synthesis will happen. So this chromosome will be duplicated, or the DNA will be replicated, or the chromosome is now duplicated into 2. So before the DNA replicates in this gap phase, the cell pretty much determines that everything in the cell is okay to go into the DNA replication mode. If it is not, there are certain checkpoints, which we're not going to talk about in this first part, and we're going to talk about in the part 2. The cell is basically assessing that if there is something not right, it will stop right here at G1 and it will not go into the S or the synthesis phase, and until the condition is made right. So that's basically what is happening in the G1 phase. Now, the next one is the S, or the synthesis phase, which is the DNA replication. The DNA will be duplicated. And from this S phase, it will again go into another phase, which is a gap phase or G2 phase. This is also very important. So here what happens, the cell will actually assess that after the division or the duplication of the DNA, whether the DNA replicated properly, whether the DNA duplicated properly, because many times during the replication, there could be DNA breaks, there could be mutations, there could be damage. Still, we do get mutations, of course, but still in the G2 phase in between, or the transition in between S to G2 phase, it will be checked that what is going on and if the DNA is replicated okay. If the DNA properly replicated, then from the G2, the cell will go into the mitosis. And this is basically the whole process. And in the mitosis, we know that there are several of the stages, and it will divide the cell. And again, that's the overview of the cell cycle. And what we're going to talk about, that first in this part particularly, this is from your chapter 18. If you remember that from the last part, that when we were talking about the microtubules, we did not talk about that 18.6, that particular area. And here we are going to mainly talk about that how the microtubules help in the process of the mitosis mainly, and how the chromosomes are separated. We're going to talk about that. And then we are going to talk about the incidents that's going to happen in the G1S and the G2 phases, particularly the checkpoint and the biochemical events, and how this is controlled in the second part, which will be on Thursday. So let's take a look at how we are going to see this. Today, we are going to talk about the mitosis, which is the part 1. Then we are going to talk about the interphase, which is a much longer. Mitosis is much shorter. If you see from, compare the two different phases, M phase is much, or the mitotic phase is much smaller than the interphase, or the duration is much smaller. So we're going to only talk about this today. Then Thursday, we're going to talk about the interphase, and the next 2 weeks, we are going to talk about the stem cells in part 3 and the cloning. And again, cloning means like we're not going to talk about the recombinant DNA, but it's a whole cell cloning. That's what nuclear transfer and those very interesting areas that we're going to talk about in 3 and 4. All right, so let's part with the real thing. So here what we are seeing, that in the mitosis, the first thing what happens, that when the cell comes out from the interphase, the cell will go into a condition where the chromosomes in the nucleus will get condensed. What do you mean by that? Or actually, I take it back. So when it comes out of the interphase, you will have this granular chromatin in the nucleus, which is not yet chromosomes. You have the DNA, they are elongated, very, very hydrated, loosely attached, and we call them chromatin. But in prophase, these chromatins, or the DNA, is going to condensed and wrapped around the nucleosomes, and it's going to just wrap and wrap. And these coilings will happen where you can see or visualize these chromosomes. Along with that, what happens, that your mitotic spindle poles will form. And how this is formed, that generally in the cell in the interphase, you will have only one mTOC, or the microtubule organizing center that you already know. Usually there is only one. Specialized cell may have many, like if you have a cilia, flagella, you all know that, that's separate. But those mTOC, or the mitotic or the microtubule organizing center, is going to also divide and move towards the opposite pole, forming the spindle pole. During the metaphase, what's going to happen, all these condensed chromosome is going to line along with the equatorial plane. Now, again, remember that this is a 2D structure. Equatorial plane does not mean that they are on the surface. Think about that. It's a 3D structure. They are going to be somewhere in the middle of the cell. That's basically what we are talking about. And, or the middle plate of the cell in the 3 dimension. So the next part, what happens in the anaphase, these chromosomes are going to be pulled over on towards each pole. Now, remember that each of these chromosomes were duplicated. So each of these chromosomes have now 2 individual chromosomes as such, which we call as a sister chromatids, duplicated. So say, for example, what will happen if we just think about that? The first one, suppose this is your chromosome number 1, and you will have 2 of them, and the other pair will also have 2 of them. So again, chromosome pairs. Now this chromosome 1 will move 1 on this pole, and the chromosome 1, the other one will move to the other pole. So each pole gets one of these. So it's an equal division, and they will go into each pole. And then next part is the telophase, where the chromosomes will decondense. They will again form the chromatin, and they will loosen up, and the nuclear membrane is going to form. So one of the important things that I just did not mention here, that once the cell goes from the interphase into the prophase, the nuclear envelope is going to completely degrade it. So that is very important. Otherwise, the mitosis will not proceed, and we will explain that why it happens. So there is no nuclear membrane here during the metaphase, anaphase, and they will start forming in the telophase, and the condensed chromosome will loosen up. The cells will again just move apart, and then there will be a pinching of these cells, which we call as a cytokinesis, where the cell division actually occurs. And we are not going to talk about the cytokinesis part though, but mainly the movement of these chromosomes using the microtubules will be the theme for today. So let's take a look at detail. That first we are looking at the interphase. As we said, the cells spend most of the time here, where you have these loose chromatin form of the DNA in the nucleus. The nucleus does have the complete nuclear membrane. And at this stage, what's going to happen, that in the microtubule organizing center, where you have the centrosome, this centrosome is going to get duplicated at the very late part of the interphase, and it will start moving towards another pole, or they will just move apart. So the next part, which is the prophase, which marks the first part of this mitosis, you can easily see that now the cell do have 2 microtubule organizing center. The centrosomes duplicated, moving towards each pole of the cell, and the chromosomes are getting condensed at this point. And gradually you will see that the nuclear membrane will also degenerate at this point. So then the important thing that what's happened, that as the chromosomes are condensing, there will be a certain region that is being developed into these condensed chromosome, which we call as a kinetochore. And that is the main thing. We are going to talk about this kinetochore constantly. This is a certain area, mainly in the middle part of the chromosome, which is highly specialized structure with hundreds of different protein. It's extremely complex, and still probably we don't know that fully. But anyway, that's going to form in the middle portion, and that kinetochore will bind to the microtubules. Now you know that why it is important. The kinetochore will bind to these microtubule, which will pull them towards each pole. So that is very important. Now remember that why the nuclear envelope is broken down. If you don't have the broken down nuclear membrane, there will be no interaction with the kinetochore of the chromosomes and the microtubule. That's the reason that they needed to be broken down. So in the prometaphase, what is happening right here, you can see that you have these duplicated chromosomes, each having 2 sister chromatids. They are going to be arranged, or trying to arrange, along with the middle portion of the cell. And this is happening by pulling and pushing. Both of these forces are occurring. And this pulling and pushing there's a particular term for that. This is known as the biorientation of the chromosome. So these chromosomes are captured by the microtubules in this stage, and they're bioriented and brought to the spindle equator. So that is very important. So you can see that here, the fully developed spindle, and all these chromosomes now are bi-oriented. They are in the middle portion of the cell. This is called the typical biorientation structure. And where you have all these chromosomes, this area is known as a metaphase plate. Because think about that although we are looking at this in this 2-dimensional picture, this is basically a plane, think about that. You have a soccer ball, and if you just cut it in half, you have this flat surface that is completely circular, and that is basically the area or the plate kind of structure where all these chromosomes align. So this is basically the middle portion of the cell, and you can see these brilliant pictures that how they are arranged on the spindle fiber. So you can see that all the chromatins, this is a DNA specific dye, so all the chromatins and the chromosomes are in blue and the microtubules are in green. They are arranged in the metaphase plate. So next, what we are going to see that in the anaphase now, these chromosomes are going to get pulled towards each other. So what you are seeing that here is an extra word here. APCC is activated and cohesins are degraded. So we are going to talk about this in the next part, but I'm just telling that what is exactly happening. So before going to this, let's take a look to the previous slide once more. So what happens up to the metaphase plate, you can see that each chromosome does have these two sister chromatids, and they are secured to each other by a kind of securing. The protein name is called a securin or cohesin that keep these two sister chromatids bound against each other. So think about that you have just two separate things, or a bundle of wood or two pieces of wood that you just secure them with something like a rope. It's just the same thing. So at the beginning of the anaphase, what happens, this cohesins, another one of the protein we are going to talk about is a securin that is a part of the cohesin, which were keeping these two sister chromatids together. They are degraded by the proteasome. So this APCC is basically a protein which will ubiquitinate a protein called securin, which is a part of this cohesin. So just pausing for a moment, it's a little bit detailed for here, but it will make sense as we talk about this in the next part. So for the protein in the cell to be degraded, if there are many different ways. One of the ways that if you just add an ubiquitin to that protein, then it is already marked and it will be degraded by a multi-protein enzyme complex called proteasome. So APCC is basically a ubiquitin ligase. What it's going to do, that it's going to add this to this cohesin proteins, this ubiquitin, and that cohesin is going to get degraded. Just think about that by the help of the proteasome. Just think about that, as I said, that you have that bundle of woods, just two of them, and you have a rope, and you have a scissor or a knife that you're just cutting that out. And that's basically what is going to happen right here. So APCC is basically cutting that rope or the binding material out so that they can separate. That's basically what is happening. So then in the anaphase A part and the B part, there are some specific things happening. And again, there is no separate anaphase A or B, all happening together. But we are, for the purpose of our understanding, we discuss them separately. So in anaphase A, the chromosome moves towards the pole. And then in the anaphase B, the spindle poles also will move farther. So it will stretch the cells. So these are the two things that is happening. Again, repeating, anaphase A is mainly the movement of the chromosome towards the pole, and the spindle pole are going to separate or elongate even farther. That's happening in the anaphase B. So in the telophase, you can see that compared to the anaphase, due to the spindle pole separation, the cell elongated much, much longer, and the chromosome reached the middle portion of each of the cell. Now you can see that at this point, a contractile ring will be formed in the middle portion of the cell. And now you know that this is done by the microfilament. Mainly, anybody want to talk to me?
Yeah, we can hear you.
Okay. No, I mean, like, I just got a call from someone. That's why I'm just wondering. Anybody does have a question so far? No? Everybody good? Okay.
I think we're good.
Yeah. Any questions so far?
I think we're good, Professor.
Okay, perfect. Yeah, just give me sometimes a feedback because it's just kind of like a trying time. So sometimes I'm just got a message from somewhere, like to call. It could be accidental. No worries. All right. So now you have this contractile ring formed, and this contractile ring is the microfilament actin, and it's going to pinch off the cells. And you can see that the nuclear envelope is being formed right here. And now you have the whole of the nucleus and the chromosome from the condensed format. It will just going to absorb or get hydrated, and they are going to form the chromatin and separate in two cells. So that's basically the portion of your regular mitosis in a little bit more detail that you have all seen in your regular Bio 101 class as such. Now we are going to see that what is going to happen after this. Now you have your first clicker question. Let's see how this works. All right, so you have, all you can see the questions, right? Can you guys hear see the question?
Yes. Yeah.
Yes.
Okay, great.
Yes, sir.
Yeah. So I just put just only 3. That's it. So cells spend different amount of times in different stages of the cell cycle. So in a hypothetical mammalian cell, a typical hypothetical mammalian cell, under all normal conditions, in which of the following stage a cell will spend most of the time? So you have to visually understand that what is the different stages and kind of assume from the previous slide where it should spend the most. Is everybody good? Anyone not okay so far? I mean, like, if you're not done, you can just tell me by unmuting your microphone. But if everybody is good, everyone? So yeah. I'm stopping it now, about 2 minutes. All right. So 80%, which is the A is the correct answer. And if you, okay, all right. Yeah, so A is the correct answer. And why A is the correct answer? Because as you can see that this is your interface, and the interface, as you can remember from the previous one, that the cell spends in the interface the most of the time. So this depicts the interface. A couple of you, about 12%, answered the pro-metaphase. Metaphase, the condensation of this chromosome actually happened pretty rapidly. So remember that your metaphase, then, your prophase, then your prometaphase and the metaphase, all these prophase, prometaphase, metaphase, anaphase is not shown. All of these are the part of your mitosis. So B, C, and D, this is happening very, very quickly. This is very dramatic movement of the chromosome, and this happens very quickly. You can visualize this live in a live cell. Actually, it happens just within most of the mammalian cell. This will happen less than 1 hour, about 40-45 minutes. This will happen, whereas this can be from hours to days. A normal average mammalian cell, if you just think about it, divides every 20-24 hours in an actively dividing cell, whereas the interphase, it will spend probably 19 to 20 hours, 19 hours in interphase in a 20-hour division, and 1 hour this mitosis will be done. So this is very, very dramatic. All right, so let's move into the next one. All right, now we are getting into the detail of that, what is happening during this particular separation of the chromosome. We are looking at what is called as the mitotic apparatus, and we are looking at the metaphase. That's important. We are looking at the metaphase, and remember that the chromosomes are now aligned in the middle portion of the cell. So the boundary of the cell is not shown. You can think about that boundary is the basically the whole slides is the boundary. So one end. And let's take a look at what are the different things that we are seeing in here. We will go gradually. We'll spend a good amount of time on this slide. So each end of the mitotic apparatus, you have the pole, which is the centrosome. This is also pole or the spindle pole. You can call it anything. So this is the centrosome, in one end. This is also another end. So from here, what you are seeing that there is these purple or pink, this is more of a dark pink colored microtubules, which is emanating from this structure, this football shaped structure, although there will be many. These are known as the kinetochore microtubules, very specific microtubules, kinetochore microtubules. The function for those that they will bind themselves with the kinetochore of these chromosomes. That's the main purpose of these kinetochore microtubules. Now remember that what we are saying that these are different microtubules, they are all the same. They are all made up of the alpha tubulin, beta tubulin. Structurally, they're the same, just they're functionally different. But other than that, they do not have different kind of structures. They are all the same, just functionally different. So the next one, what you have is these dark blue or these purple shaped structures. These are known as the polar microtubules, polar empties, polar microtubules. So they extend from each of the spindle pole towards the opposite pole, but they don't touch or attach to the kinetochores, or they do not have anything to do with the chromosomes. So they do not touch the chromosomes. And all of these are happening randomly. Remember that when the microtubules are growing, it is not that it is targeting to touch the kinetochore or is targeting to bypass the kinetochore. It is just randomly the ones which will touch the kinetochore and attach to them, they will be named, or they are distinguished as kinetochore microtubules, which will just bypass the chromosomes and just grow towards each other. They will be called as polar microtubules. And then you have the last one, that from the pole, there will be a bunch of microtubules which will extend towards the plasma membrane or towards the periphery of the cell, which are in the yellow. These are called the astral microtubules. And this is actually responsible for elongating the cell or making the cell bigger. And we're going to talk about each one of them. So one of the important things that you have to understand from here, remember that all of these microtubules do have a very specific orientation. So the spindle pole, or towards the spindle pole, all their minus ends are directed towards the spindle pole. So away from the spindle pole, if they are going towards the periphery of the cell, they are plus-ended, The kinetochore microtubule, which are growing towards the kinetochores or towards the middle part of the cell, they are plus-ended, where the minus end is towards the spindle pole, and the same thing for the polar microtubules. So everything close or towards the centrosome or the centriole or the spindle pole, whatever you call it, they are all this minus orientation, or this the minus end is towards that. So that's the important part that we're going to talk about. What is the important thing is right here, what you're going to see, that what does it mean by the zone of interdigitization? So the interdigitization means that if you just look at these polar microtubules particularly, they will pass against each other. And in this particular zone of interdigitization, these opposite microtubules or polar microtubules are going to interact with each other with the help of motor proteins. And that is actually important. So that's the reason that we call them as a zone of interdigitization. And this interdigitization is only going to happen, the interaction is only going to happen with the polar microtubules. Remember that interdigitization will not happen with the kinetochore microtubules, and explain that why it is so. So let's take a look at what we are seeing in this particular picture. You have two sister chromatids. Each of them do have these green kinetochore. So each sister chromatid has one. And if you see that there are, like say, for example, two of these kinetochore microtubules, those are attached to the kinetochore on this sister chromatid. There are two microtubules from the other side pole, they're attached to this sister chromatid. So this is very important that, again, I'm repeating slowly this, that the kinetochore, only the kinetochore microtubule from one side of the spindle pole is going to attach. And this will cause what is called the biorientation of the chromosome at the metaphase plate in the middle portion of that. If there is suppose I'm just saying that if that happens, you have a particular kinetochore where you have the microtubule attached from this end, and the microtubules are going to attach from this end. At that point, what will happen that when you have the anaphase, that chromosome cannot move because each one of them is trying to pull that chromatid towards this pole, and that will really not happen. So when you have the particular microtubules from towards one pole, and this is from the other pole is attached to each other, during the anaphase, when they shrink, and we'll explain that how it happens, they will be going towards the opposite pole. So that's very important, that each kinetochore can only bind to the microtubules from an individual spindle pole, either this one or that one. So that's basically the structure. And by the help of this particular attachment of these microtubules or the kinetochore microtubules, these chromosomes will be bi-oriented. That means that they are attached by the opposite kinetochore microtubules. This is at the metaphase. So let's take a look at this in a little bit more detail, what is happening. So here, before going into the detail of the structure, let's take a look that these are the two sister chromatids. Now, if you remember that, we talked about this cohescence. Now it makes sense. As I said, that think about that two pieces of wood attached with a rope or bundle. This is exactly these cohescent proteins. These are keeping the two sister chromatids together. But what will happen, but we just talked about that during the anaphase, as they have to move apart, the APCC complex will degrade these cohescents so that they are separate and they can move together. So now let's take a look only at this portion, which is the centromeric region of the chromosomes, which is known as a kinetochore. So here you are seeing the microtubules are attached to the kinetochore of each one of the sister chromatids. You have kinetochore 1 and kinetochore 2. So each one of them have their own. So what are the different structures here? So what you are seeing that this is the kinetochore, which is also known as the outer kinetochore. This is attached with the plus end of the microtubule somewhere. And again, remember that this is an extremely large protein complex, although shown just here, just as just one unit. The next portion is also known as an inner kinetochore, which also contain a specialized type of DNA, which is known as a centromeric DNA. So we're going to talk about that. And this is the body of the chromosome that you are seeing. So here in an electron micrograph, the greens are the kinetochore microtubules. The outer K portion is your outer kinetochore. This C is the centromeric DNA in the inner kinetochore. And this dark blue is the chromosomal DNA. And again, this is pseudo-colored black and white picture of a transmission electron micrograph, but just colored that way. So this is the overall structure. Now let's take a look in a little bit more detail, and don't get intimidated by this picture. This is just for an appreciation of that, how complex this structure could be. So if you try to orient that, if you have this structure sideways, now in the next picture, the structure is upward. That means that this is coming from the top. So you're looking at, so we are flipping this structure 90 degrees, that's what you are seeing. So think about that. Now this kinetochore, this is the region, and these are the kinetochore microtubules coming from here. And there is a region right here outside the outer kinetochore, which is known as a corona, unfortunately or fortunately. So that portion, so once that is traversed, you can see that the microtubules are attached to the outer kinetochore right here. And then you have the inner kinetochore. All I'm pointing out here that this centromeric DNA is around these histones, and there is a very specialized type of histone here, which is known as a histone 3. That's the only name that you need to remember. histone 3 are a very specialized type of histone, and that marks the inner kinetochore. You will not find that histone 3 anywhere else. And that, since histone 3 is going to get replaced by another type or another variant, which is known as a CENP-A. Centromeric protein A is also a histone, which is only found in this inner kinetochore. So why this is so? Because this makes this region very special for a particular kind of association with the microtubules. And along with that, so only thing that I'm pointing out is this CENP-A nucleosome, which is the only name that you require. And another thing that we will talk about much later is this ORB. This ORB stands for an Aurora B kinase that we are going to talk about, and that is also responsible for this attachment of the microtubules and the movement of the chromosome and the interaction with the corona, outer, and the inner kinetochore. So really nothing much is required from this, just rather than that how complex that system is. And we're just looking at this for the purpose of appreciation mainly. So let's take a look at the next question. All right, so now you have, all right, which of the following diagrams correctly depicts the orientation of the microtubules during the metaphase of mitosis? About 1 minute. Is there anyone having difficulty? You can unmute and talk if you're not done. Good, everyone? The reason that I actually can see, because there are 98 participants and I have 93 response. So I'm just, that means 5 less. So good, everyone? I'm stopping it now then. All right. Yep, so you can see that D is the correct answer. That is correct. So we saw before that, just remember that always you have the spindle pole and the minus end of the microtubules are always towards the spindle pole. And from there, these are the astral microtubules towards the periphery of the cell. They are all plus ended towards the periphery of the cell. Then you have the kinetochore microtubules, which are attached to the kinetochore. They will attach with the plus ends. And then you have the polar microtubules, which are bypassing the chromosomes. They will also have the plus ends towards the middle portion of the cell. So that is the correct answer. See right here, these are not the minuses as well. Yeah, so that's basically what it is. All right, great. So let's move on. So the next thing that what we are trying to see, that we already saw the kinetochore microtubules. So the next thing is that how these chromosomes are getting attached to these microtubules. And this model is known as a search and capture. Now, really, search means that it is a chance event completely. So during this late prophase and during the metaphase, all these microtubules are growing randomly from each pole or the spindle pole towards the middle portion of the cell and towards the outside of the cells. They are just growing and growing. So what will happen that in one case, if by chance a microtubule attaches its end towards the kinetochore, it will have an interaction. And this is known as the end capture. And again, remember that this is randomly happening. They are growing, shrinking, everything happening all together. Other one is that what can happen that if the microtubules are just kind of brushing off, not away, remember that if they are away from the kinetochore, they are good. This is a very important concept. If they are not within a very close distance in between the kinetochore or a protein complex, remember that if the proteins need to interact, they have to be almost close to less than 10 nanometers. Otherwise, they're too far apart to interact. If they're too far apart, they are going to be polar microtubule. But if they are kind of a brushing by the side of these kinetochore, they can also interact with the side, and this is known as a side capture. But they have to physically touch the kinetochore by the side, not just passing by. So that's the important concept. So they can either be captured by the end capturing or the side capturing method. And another important thing, that always remember that you are seeing these two sister chromatids, each have these kinetochores, and each kinetochore can only bind to one set of kinetochore microtubule emanating from only a single pole. So only these microtubule from this single pole can bind to this, because as it is binding, no microtubule from the other end can bind to this. It can bind to the other one, but not this one. And this is maintained by some specific processes, and we're going to see that as happening. And that will help in the bioorientation of the chromosome during the metaphase. So let's take a look at what is happening in a little bit of detail, which is called the chromosome capture and this congregation in the metaphase part. So the congregation is basically they are all bunching towards the middle portion and trying to be on the metaphase plate in the next portion. And this is known as a congregation of the chromosomes. They are just kind of oscillating and attaching, and they're trying to move towards the middle portion of the cell. These are visually very striking. You can visualize them in the live cell, actually. I will just try to post some videos. So let's take a look at what is happening here. So in the 1A, you can see that there is a kinetochore microtubule attached to the kinetochore, and this is trying to grow in this direction, in the sense that this is the plus end, this is the minus end, and this is having a tug of war in the sense that the other side is not attached to anything. So it is trying to go towards this direction. And I will explain that how this is happening. On the 1B, you can see that there is a side capture of one chromosome. On the other hand, see that what is happening right here. In this, there is a side capture of this, and this chromosome is trying to go on this side. On the other hand, see that here is a side capture of this chromosome, which is going to go on towards this side. So here it is attached to one of these chromosomes trying to grow on this side. So what is happening right here? So let's take a look at what in detail, that why these different things are happening, which is called the chromosome congregation, after which all of them end up in the metaphase plate. So if we look at the chromosome in detail, you have these two sister chromatids. Right here, you have the kinetochore. They are still bundled up with these cohesins together. And on each side, you have these kinetochore microtubules. So this is a kinetochore microtubule. You have the plus end here, minus end is towards the spindle pole. You have the plus end here, you have the minus end towards the spindle pole. So during the congregation, what you are seeing, that suppose this is one of the kinetochore that is attached to this kinetochore microtubule. And this is attached with this. And here, what it does, it recruits. Now you know that from your previous knowledge, this is your dynein-dynactin motor complex. So this dynein-dynactin motor complex is attached to the kinetochore protein complex, and it is sitting on this particular microtubule. Remember that your dynein will try to move towards the minus end. So now this is trying to move towards this minus end. This is one thing happening. On the other hand, if you clearly see right here, there is also another protein right here, kinesin 7. And the kinesin 7 actually are kind of a dual head. That means that it does have a head on each of the end. One of the head is attached to the kinetochore, the other one is on the microtubule. And the kinesin 7 is hanging on there because remember that kinesin is a plus-ended motor, so it will try to go on this side. Dynein is going to try to go on this side. Kinesis is trying to go in this side. So it's kind of a messed up situation for this chromosome. On the other hand, see that, let's see what is happening right here. On this one right here, this chromosome is actually shrinking. This one is growth. This one is shrinking, and this shrinking is due to this kinesin 13. If you remember that, they can bind to the plus end of the microtubules and bend the microtubules, and you have a disassembly. So there is the disassembly towards the plus end. And here also you can see that you have a dynein motor complex the same way, but the difference is that this kinetochore microtubule attached to the other sister chromatid kinetochore. So this one to this sister chromatid, this one is to this sister chromatid kinetochore. So what is happening at this point? So as this is shrinking, this dynactin and the dynein motor complex is trying to walk against towards the minus end. So this sister chromatid is trying to go towards this end. On the other hand, here, this dynactin motor complex is trying to pull it towards this end. On the other hand, this kinesis 7 is trying to move it towards this plus end, and see that there is growth. So this is actually growing, and kinesin to some extent wins to some extent because it just hangs on and tries to grow towards this end. So you can see that all these cause these oscillations. It's trying to go this way, it's trying to go this way. And finally, what happens by the growth of this and shrinkage of this, it is ultimately all these chromosomes end up in the middle portion. And this is done only due to this, because if this is not shrinking and if this is not growing, they will not be able to end up in the metaphase plate. And why this is happening? Because still both of the sister chromatid are attached to each other. You have pulling, pushing, both going on this end. You have only pulling going on this end. But again, ultimately, you have the winning, and this kind of like a, although it's kind of funny for just to picture, think about that. You have one dynein here, you have one kinesin here, so you have two of them trying to push this chromosome towards that. And here you have just only one dynein here, so you have 2:1, this loses, and this chromosome moves towards the, like shown here, moves towards the middle portion of the pole, and it will just attach, it will biorient itself in the middle of the pole. So this is basically what is happening during the late prophase and in the pro-metaphase, and at the end of the metaphase, they will all line up together. And
how does it know? How does it know when to stop in the middle? Like, why doesn't it just keep going?
Yeah, so a very good question, actually. So the thing is that it is just an equilibrium. And if you just read the text in your book, it is basically a random orientation, and it will just oscillate in this middle portion. And it's just an equilibrium. That's basically what it, due to that. And what happens at one point of that equilibrium, what will happen that these will break out, and at that time it will be pulled towards this end and it will be pulled towards that end. So that's basically the overview of that. So that's basically a kind of and again remember that each of the kinetochore is going to be attached to hundreds of individual microtubule, and the force distribution and the motor protein association is kind of going to equal out. So that's basically the equilibration of any biochemical reaction, that how it happens, and that's basically what it is, because the overall process is both sides is going to be almost the same. Does it make sense?
Yeah, thank you.
Yeah, sure, yeah. All right. So let's go into the next portion. So here what we are seeing, a little bit more detail of the same region. So here you have the chromosome right here at the back, and right here, this portion is the A on the centromeric chromatin that we talked about, which is the variant of that histone 3. And right here is the inner kinetochore. And this is attached by these long filament thread-like structure to the outer kinetochore complex. And this outside, you have this NDC-AT complex. And this is a very, very complex protein. Hundreds of proteins are together in this NDC complex, which is known as the nuclear division cycle AT complex. But again, that's not needed. But just the outer kinetochore is attached to the inner kinetochore by this fashion. So this is a very interesting thing, and why this is required. This is just like a ring. You are only seeing that the one end. So this is like a sleeve and the ring, and the inner kinetochore is attached to that. Because remember that we talked about that there is addition, and also at one end, which is the the other side of the kinetochore microtubules, you will have degradation or depolymerization. And someone asked this question before, that if it is attached really to the end, if it grows or if it shrinks, what happens? So see that this end is actually not interacting with anything. It can bind or it can grow or shrink, because this is the sleeve through which it is attached. And the motor proteins can work right here to push or pull this on this part, whereas easily as it grows, it can go to this end. As it shrinks, it can go towards this end. So this is basically the machinery of the outer and the inner kinetochore, how it helps the biorientation of each one of the chromosome. So if this is on this side, you will have another microtubule on another sister chromatid on this side with the same kind of orientation. And this structure is an extremely rigid, very, very strong structure. And we're going to see that how this helps in the separation of the chromosome after the biorientation at the metaphase. So let's take a look that right here, we are going to look at that how this is regulated, or the biorientation of the chromosome is regulated. Let's see if we have the additional one. So looks like my pointer is stuck. Okay. All right, now we have the red one. So right here, what we are looking at, now you have these two sister chromatids, and they're secured by these cohesin molecules. And what you're looking at right here is your inner and the outer kinetochore. And what thing is I want to point out that all these red proteins, these proteins are riding onto this centromeric region of the chromosome, and this is known as the CPC, or the chromosome passenger complex. And this contains a kinase which is known as the Aurora B kinase. And again, remember that all the kinases phosphorylate other proteins. So that's basically what it does. So let's take a look at what exactly is the role of these Aurora B kinase. So here what you are seeing, that prior to your congregation of the chromosome, like when it is in the prophase state, at that stage, you can see that inner and the outer kinetochore do not have much tension, and the Aurora B kinase can phosphorylate all of these proteins. And you can see that these are the phosphates. They're all phosphorylated. And at this time, what happens that even if there are some microtubules coming and touching them, due to this phosphorylation of these proteins, there is a weak interaction, and this causes a low tension. Now, what happens that as the cell is going from the prometaphase towards the metaphase, more and more microtubules are growing. So just the amount. Initially, it was just low, and now it is growing and growing. So what will happen that some of these microtubules will be associated with the inner outer kinetochore, and it will try to pull the kinetochore towards itself, as you know. But again, as the chances of each of these kinetochores to be pulled are increasing, you can see right here, both the chromosomes are still attached, but this middle portion is going to get separated. And there is a tension being built up. This is just by the amount of microtubule going and attaching to the outer kinetochore. So at that time, what happens, due to the tension buildup, these Aurora B kinase become too distant from these outer kinetochore complexes. Here, they are very close. Now, the distance is elongated. So you can see that here, this is the distance. Here, the distance increased to some extent. And due to this, you can see that the Aurora B kinase is kind of no longer can go or reach this NDC80 complex. Remember that the NDC80 is the outermost complex, or the NDC80 complex will be within the distance of the Aurora B kinase. And with the phosphorylation, it will not be able to pull or develop this pulling force that much. What happens that when it becomes dephosphorylated right here, the Aurora B kinase, So here there is no Arabic kinase because due to the development of these forces. As more forces develop, there will be more of dephosphorylation of the NDC complexes. And basically, this helps in the bioorientation because without this phosphorylation, the attachment of the NDC-AT complex to the microtubule are much stronger. And this is a high tension, and this is pulling the two of the kinetochore regions apart. And at some point of time, these cohesins will also degrade so that they are pulled apart as well. So now let's take a look at how exactly the mechanism happens right here. So right here, although when you have the metaphase, remember that in the metaphase, in the middle portion of the cell, all of these chromosomes lined up in the middle portion of the cell in the metaphase plate. And looks like that at that point, this was also that question, another interesting thing, that looks like that the microtubules are neither growing nor shrinking, which I told that this is an equilibrium state. So it looks like that, but there is constant treadmilling going on. That means that there is addition of the tubulins towards the plus end, and there is a subtraction from the pole. And this happens in both of the poles, on this side of the spindle pole, this side of the spindle pole as well. And this kind of equilibrates these conditions. So that's the reason that due to the equilibration of this subtraction and the additions are the same rate, they kind of line up in the middle portion of the cell. And both happens that both of these on each side of the chromosomes happen at the same rate. So you can take a look at this, that you can just add a fluorophore to these tubulin individual subunits, alpha, beta subunits. You can add a fluorophore, and you will be able to see that they just load on one end, the plus end, and they will be treadmilled towards the minus end. And that experiment can be done as well. So let's take a look at the next part. So that's all about your metaphase. Now in the anaphase, anaphase A and the B, although we separated these two, but remember that both of these are happening at the same time. So right here, now you have already seen that these kinetochore microtubules are going to pull the chromosome towards this end, this end, and this is happening due to these motor proteins walking towards the minus pole. And why this will separate, remember that we talked about that the cohesins and the securins, which it was attaching the two sister chromatids, are going to get degraded so that they can be separated as well. So this is what is going on with the kinetochore. Along with that, there are two forces that is happening to elongate the cell. That is the part of the anaphase B, as we depicted. So one thing what is happening is in the polar microtubule. So here you can see that overlapped polar microtubule, they are going to push each other. And how they push? They push by a dual head kinesin, and we're going to see that. Some motor proteins are going to push this microtubule towards each end. That will make the poles move farther. The second thing that is going to happen, that these microtubules, which are the astral microtubules, these astral microtubules are attached to the plasma membrane, or they're touching the plasma membrane rather than attaching. And there are some dynactin motors which are attached to the plasma membrane. They are trying to walk towards the minus end, from the periphery towards the minus end. That makes a huge tension, because remember that this is the minus end. The plus end of the microtubule is trying to push the plasma membrane, whereas the plasma membrane is attached to the dynein motors trying to walk towards the minus end, and that tension elongates the cell. Now, the next slide will make it much easier to look at. So let's take a look one at a time. So what we are looking, this is the overlap zone of the polar microtubule. Right here, you have a kinesin. This kinesin is attached to this end, and remember the kinesin moves towards the plus end, and here it is attached to this plus end as well. And this will push this way, and this will push it this way. But this picture is a little bit more of a kind of a older picture, because in reality what happens that these are actually dual headed. So a head, we are not saying, although I depicted or said that this is the tail is attached there where the head is pushing it this way, whereas this head is trying to move it this way and the tail is attached there right here. That's the reason they are pushing. But in reality, the tail also is a head. So we're going to see that in the next slide. So this head is going to try to move in this way. This head is going to try to move it this way, and they are pushing each other, the poles apart. On the other hand, this is your plasma membrane, and the dynein dynactins are attached onto the plasma membrane. And as the microtubules are growing, it grows towards the plus end. This dynein dynactin is trying to move it towards this end. This makes the tension, and the plasma membranes are pushed farther apart. And also there are some evidences that in the middle portion of these polar microtubules, there are also, so to the plus end, there is polymerization of new tubulin. So again, as the new polymerization of the tubulin happens, the length of the microtubule increase. That also helps in the lengthening of the whole cell in the anaphase. So that's the overview of that. So let's take a look at that at the molecular level. So the first thing what is happening is, let's take a look at how the chromosome is being moved. So here you have the chromosome, and this is moving towards this end by the help of dynactin motor. Remember that it just moves, not shown here, but remember that the kinesin 13 is shrinking it from the minus end, and the dynein dynactin motor is trying to move it towards the minus end. That will pull this chromosome towards the center. That's what's going to happen. Then what is happening right here? You have the kinesin 13 that is shrinking towards the minus end. And that's the reason that this is right here. It is you have a shrinking on this end, you have a shrinking of this end. All of them helps to shrink this. This happens to these polar microtubules as well. But see that this is the interesting part, which is happening at the overlapping part of the polar microtubule. You can see that this is what I was talking about. This is a kinesin 5, where you have the two heads and the tail. On the other end, you have two heads and the tail. So the tail is in the middle, and you have dual headed. So the kinesin 5 is dual headed. So in the overlap part, this kinesin 5 is binding on this. This is bound on this. Remember that kinesin always moves towards the plus end. So this head is trying to move it towards this end. This is trying to move it towards this end. So this basically, this is a tug of war. So it's going to push this microtubule towards the minus end. This is going to move towards the minus end. And as it is happening, you can have more of a polymerization towards the plus end as well, as we saw in the last one. So that will elongate the cell, or the spindles will move farther apart. And this is happening at the periphery of the cell. So here you have your dynein dynactin motor complex attached with the plasma membrane, and that is trying to move towards the minus end. So from here, it will try to move towards the minus end, but there is polymerization of the plus end is going on. So the microtubule is growing on this end. So in this condition, you have a tension that will push this plasma membrane even farther due to that, and that's how it will elongate the cell. So last clicker question. So let's take a look. So which of the following does not facilitate the chromosome separation during anaphase of mitosis? Microtubule polymerization, microtubule depolymerization, kinesin 5, dynein-directed forces. All of these facilitate chromosome separation. It's a tricky question, though. Do you want to change your answer, thinking about it a little bit? All I can say is that E is not the answer, though. then choose from A, B, C, D. What happens? Remember that congregation, when you have the chromosome congregation at the pro-metaphase, and after that, it is pushed and pulled towards the middle portion of the cell. But here the question is that the separation during the anaphase of the mitosis, separation, not the cell that is increasing in size. So that's your clue. Not the cell that is elongated, or not the cell that the spindle poles are pushed apart, only the chromosome that is moving towards the spindle. Good enough time? Good. Anyone having difficulty answering? Almost 2 minutes. All right, I'm stopping it. So 46% answered A, and A is the correct answer. So microtubule polymerization. See that this is a difficult question. So again, once you read the material, you will probably get it. So let's take a look here, and a couple of different things going to look at. So here, if you carefully look at that as I said, that here you have a polymerization. That means during the congregation, during the pro-metaphase, this is polymerized. But see that what is the consequence of that? This polymerization will push the chromosome towards the middle portion of the cell so that it is bioriented on the metaphase plate. It is not separating the chromosome. It is putting it in the middle portion. So on the other hand, what you're going to see that the chromosomes can only move towards the pole due to the shrinkage, not by the polymerization. Now, let's take a look here. You can easily see that once the chromosomes are, so here you can see that you have the polymerization, but these are polar microtubules. So even if there is polymerization in this condition, or you have polymerization here even, these are not attached because this is anaphase B. They are not attached to the kinetochore microtubules. So here what is happening, that when these chromosomes are pushed apart, see that here you have this kinetochore microtubules attached to this kinetochore microtubule attached to this kinetochore, and here see that what is happening, you have shrinking of the microtubules that is pulling the chromosome towards the pole. So does that make sense? If you have a difficulty, I will explain it once again. Remember that. So here, think about that. This is the condition. You have the shrinkage. Remember that the motor proteins are attached to this end, the dyneins. They are moving towards the minus end as this end is getting shrunk. And that's the reason that it is moving towards the minus end. Remember that along with that, the minus end is also shrinking, not only the plus end. Just to make the process rapid, kinesin 13 is shrinking the microtubules at the plus end, right here at the plus end, as well as the minus end. And that's the reason that it is so rapid. So from the metaphase to the anaphase, that happens just within 5 to 10 minutes, the movement of this chromosome, very, very rapid. And that is done by this. So it is always, so that your question is microtubule depolymerization, kinesin 5 directed forces that will separate, and the dynein directed forces, and all these facilitate chromosome separation or not the microtubule polymerization.